patient are suffering from the hypertension, and then next, when I maximum number of the diabetic patient complaining about the arthritis, and there is no such case of cancer and asthma. Now come to the whole exome sequencing part. At first, we need to know what is exome. As we know, every gene has two parts, that is intron and exome. The intron is deleted during the transcriptional process. Only exomes are code for the protein in the translation. So this is required to understand is there any protein coding SNP are they are present or not to evaluate their disease prognosis. So what I have found, the diabetic sample have more synonymous and non-synonymous mutation and compared to the non-diabetic population, and diabetic samples have more number of pen-shift variation and infant variation in their exome. Here you can see number 11 to 20 are diabetic sample and number 1 to 10 are the non-diabetic sample. Then I analyze the common variants that is associated with type 2 diabetes mellitus. What is common variants? Common variants are those variants which can be present both case and control, and they do not play any significant role, or maybe benign types have slightly significant role in the prognosis of diabetes. Here we can see approximately 75% of the common variant that is shared by the both diabetic and non-diabetic population, and which is represented in this bar diagram, which shows the frequencies of some of the common variants that is shared by the both diabetes and non-diabetic variants. And here we can see the PPART RS1801282 that is unique to the diabetic population. And then I analyze the pathogenic variants. Pathogenic variants are those which is highly significant for type 2 diabetes mellitus, and if one of these kind is present, there is record the person may develop diabetes in his future, or when he may be develop diabetes in his throughout his life. Here I've seen that the 40% of the gene that is shared by the both diabetic and non-diabetic population, and we can see the frequencies of the distribution of the gene. Then I constructed a network of the gene which is responsible for the development of the diabetes and their association. Here I have found that there are multiple genes who are linked, or they co-translated and they develop diabetes mellitus, and when I ranked them, I have found that CDKL1 is the most prominent gene that is responsible for the development of type 2 diabetes mellitus in such population. Now comes to the second part, that is investigating the pharmacodynamic response of metformin in ethnic population throughout the OCD genotyping. Now we first need to understand what is pharmacogenomics. Pharmacogenomics is the study where an individual's genetic makeup influence his drug response. So for this purpose, I have selected metformin as a candidate drug. Why metformin? Because metformin is prescribed as a first-line therapy for the treatment of type 2 diabetes mellitus, and it is considered as gold standard first-line therapy for the treatment of type 2 diabetes mellitus. If we look at the pharmacokinetics of the metformin, there is an interesting thing about the pharmacokinetics, that the metformin do not metabolize in the liver, and it excreted unchanged by the kidney. So it is difficult for us to determine the pharmacogenomics of the metformin. So we have come to a conclusion, rather than to analyze the type of metabolizer of such a population we decided to analyze the metformin responders. Metformin responder means person who respond to the metformin, or non-responder means person who do not respond to the metformin. So here we have found that the OCD1 protein that is responsible for the metformin action in the liver. So OCD1 protein that is located in chromosome number 6, and the size of the nucleus is 36,904 base pair, and the protein size is 554 amino acids. So now we'll understand how OCD1 plays its role to manage the type 2 diabetes mellitus. So when inside the mitochondria of the hepatocyte, when glucose enters into the mitochondria, by the help of mitochondrial complex 1, it produces ATP and subsequently the gluconeogenesis. And when metformin enters into the mitochondria through the OCD1 receptor, it inhibits the mitochondrial complex 1, which inhibits the ATP production and which increase the AMPK, which subsequently inhibit the glucogenic enzyme PBCK, G6PS, and which is ultimately inhibit the gluconeogenesis process. But if there is a change in OCD1 protein, the structure of the OCD1, the less amount of metformin enters into the mitochondria of the hepatocyte. As a result, AMPK is inhibited, which increases the gluconeogenic enzyme like PBCK and G6PS, and which ultimately increases the gluconeogenesis. And the ultimate result is uncontrolled diabetes or glucose level. So, from the WES result, we have screened for the missense mutation that are present in the OCD1 gene. We have screened there are more than 3 genes, the missense mutation that are present in the OCD1 gene, and the rate is higher in diabetic population compared to the non-diabetic population. And from those genes, we have again screened the SNPs which are responsible for the altering of OCD1 protein. And we have observed there are more than 1 SNPs are responsible for the altering of the OCD1 gene action. And the rate is higher in non-diabetic population compared to the non-diabetic population. So I have selected those 2 SNPs. Those are RS122-08357, which reduces the OCD1 function and lower metformin uptake